Hello everyone, I'm Giuliano Zanchetta, and today I'm gonna tell you about the design and optimization of a rapid multiplex microRNA assay. Let me first thanks my co-workers from the Complex Fluids and Molecular Biophysics lab at the University of Milan, and our collaborators Carlo Morasso in Pavia and Giovanni Tagliabue from Proxentia, and Regione Lombardia for funding. MicroRNAs are single RNA strands which act in our body as post-transcriptional gene regulators. They are widespread in our body. There are 2,000 of them, and they are also circulating in our blood, and they are actively investigated as biomarkers for several diseases. Therefore, their detection and quantification is a very interesting challenge. It is a challenge also because their concentration in blood is very, very low, between femtomolar and picomolar. And therefore, there are several different techniques used for this task, like PCR or microarrays. Some of these techniques are very sensitive. Some of them are allow high throughput and high parallelization. However, all of these techniques require a furnished lab, many steps, and therefore they end up in a high cost and high time to result. Therefore, the question is, can we improve? To this aim, we are using a reflective phantom interface, or RPI, which is a label-free optical biosensing technique developed in our lab. It is based on the very low optical reflectivity from a surface, which is coated with a functionalizing polymer, which allows to add receptors on the surface, specific and different receptors on the surface, so that we have a very low reflectivity functionalized surface. If we add targets in solution, we see that the mass absorption of the targets onto the receptors results in a change in reflectivity, so that we can monitor the increase in reflectivity, and therefore we can monitor the increase of attached mass on the surface in several different spots in parallel, which allow us to estimate the kinetic parameters of binding and unbinding and the affinity of the target receptor interactions. Therefore, we can study several different couples of receptors and targets, and we have studied many of them, among which DNA-protein interactions. Recently, we have been studying transcription factors, and we can functionalize the surface with several different sequences, double-stranded sequences, bearing or not the consensus sequence. And therefore, by increasing the amount of protein in solution, we can study how it binds to several different sequences on the surface, so that we can estimate the affinity, the kinetics, the dependence of ionic strength, and we can estimate entropic and enthalpic contributions, which we compare with the results of molecular dynamics simulation. Also, we studied a lot recently interactions between DNA and DNA strands. For example, we can tune the surface density of probes on the surface, and we can study the effect of this on the kinetics. And we revealed a very high sensitivity to even single nucleotide mismatches between target and probe, so which we can estimate with the usual approach. Also, while doing this, we realized that there are several contributions to the probe-target interactions at the surface. For example, very weak, non-specific interactions between the probe and the surface can have an effect on the measured affinity. Also, we studied the effect of electrostatic repulsion and therefore the effect of ionic strength, because charged strands approach charged probes on the surface, and this can have an effect on the kinetics. Also, we characterized how possible probe-probe interactions affect and compete with the target-probe interaction. And on the other hand, we realized that changing the spacers, the length and flexibility of the spacers can have an effect on the measured affinity. So with all of this knowledge, we wondered, can we combine such knowledge into an effective way to detect and capture microRNAs? And therefore, we developed a one-pot assay based on, again, functionalization of our surface with DNA probes. And if we add a very low concentration of microRNA in solution, we see that we barely see any effect. However, if we add in solution afterwards without washing a primary antibody which specifically targets DNA-RNA hybrids, we see that only in the specific spot we see an increase of the signal. And we can amplify such signal by adding in solution, after a while, a secondary antibody, which is binding specifically to our primary antibody. So we end up with a relatively fast, less than 2 hours assay, which is combining 3 steps without washing, which allow to amplify specifically a very low concentration of microRNAs. To do so, to be able to do so, we characterized each single step of these interactions, both in terms of affinity and kinetics, which proved very critical to develop a sensitive assay. And therefore, I'm gonna tell you a bit about these different steps. So we chose 5 different microRNAs, which are biomarkers for different cancers. And we characterized the full interaction in terms of affinity between DNA probes and microRNAs in solution, in terms of affinity and kinetics. And this is very important because it ends up that the effective rate of interaction between microRNA and the DNA probe on the surface is the key element to determine the detectable, the smallest detectable concentration, which we are able to spot for each specific microRNA. Also, we characterized the action and the binding of the primary antibody to the DNA-RNA hybrid. This has been reported already several times, but we characterized it for our system, and we found that this specific antibody for hybrid DNA-RNA hybrids is very, very strong, has a very high affinity, and also it's sequence independent, and it's acting very nicely with every different sequences that we explored. And it provided a pretty good signal amplification. Also, we characterized the secondary antibody interaction with the primary, and we found that the valence, the effective valence, is higher than 1. So several secondary antibodies can bind to each primary antibody, resulting in a further amplification, almost 100-fold. So to try to model and rationalize this behavior, we modeled this behavior with a nested set of differential equations, which are describing each single process: the binding of RNA to DNA, the binding of the primary antibody to the hybrid, and the binding of the secondary antibody to the primary antibody. And since we measured each of these interactions and the kinetic parameters for each of the interactions, we can precisely emulate and reproduce the experimental results. And this gives us a tool to estimate, to explore the whole parameter space, to look for the combination of relative concentrations and time intervals, which provides the highest amplification of the signal, of the secondary antibody signal, as compared to the signal which we would obtain by only waiting for the microRNA to cover all of the DNA probes. And so we did so for a large set of parameters, setting the total duration of our assay to just 1.5 hours. And we found that the best combination of parameters is for a very small incubation time for the microRNA only, and a very long interval time for the primary antibody, which is critical to obtain a large amplification. And also, we could, by studying the effect of concentration of the primary antibody, we found the best possible amplification. So with this set of parameters, we turned back to the experiments. And indeed, we found that if we look at the signal of the final signal of the secondary antibody, we find that it is scaling linearly with the microRNA concentration in solution. And so it's providing a very nice signal. And above all, it is providing a picomolar to sub-picomolar limit of detection without washing steps, without nanoparticles, without enzymatic amplifications. Therefore, I showed you how we developed a rapid one-pot multiplex assay for microRNA detection and quantification. And I tried to show you how we could optimize and maximize the sensitivity of our assay down to sub-picomolar limit of detections, thanks to the RPI quantification characterization of each single step. And I showed you how the kinetics of microRNA DNA binding is a key factor for determining the limit of detection. So our next step, which we are pursuing, is to work on real samples, which we are extracting in order to purify our microRNA and enrich the samples and look for biomarkers in our plasma. And with this, I thank you.